Welcome to the deep dive. Today we're really getting into a fundamental chapter, crucial for class 11th and 12th boards, and of course for NEET and other medical entrance exams, plant growth and development. I'm really keen to understand how plants, starting from just a tiny seed, manage to develop into these complex structures, and what exactly controls these amazing processes.
Bahut ache. Yes, plant growth and development is absolutely a cornerstone of plant physiology. It's essential for scoring well. It tackles those really basic questions about life itself, doesn't it? How does a single fertilized egg, the zygote, give rise to a complete plant? With roots, stems, leaves, flowers, fruits, and seeds. It's quite incredible.
It really is.
So today we'll unpack the precise and highly ordered succession of events involved. We need to understand not just the what, but also the why and the how behind these plant life cycles. Let's begin by defining the core concepts. Okay, so think about any flowering plant you see around you. Maybe one you've studied before, its overall organization, its anatomy. But have you ever really stopped to think about where and how structures like roots, stems, leaves, and then later flowers, fruits, and seeds, where do they actually come from?
Not in that much detail, I guess.
And it's always in such an orderly sequence, right? It's not random at all. You know the basic journey: seed, then seedling, then a little plantlet, and finally the mature plant. Now here's something interesting. Think about a huge tree. It can keep increasing in height or girth seemingly forever, right? Almost unlimited growth.
Yeah, for centuries sometimes.
Exactly. But its leaves, flowers, and fruits, they have very defined sizes. They appear, do their job, and then fall off, sometimes repeatedly.
Right, that's a different pattern. More limited.
Precisely. It's like two different developmental clocks running. And have you noticed how the vegetative phase, the growth of roots, stems, leaves, almost always comes before flowering?
Yes, that seems consistent.
And every plant organ is made up of all sorts of specialized tissues. So is there like a connection between the structure of a cell or a tissue or an organ and the specific function it performs?
There must be, right? Structure fits function.
Absolutely. And can these structures and functions actually be altered by the plant itself? That's another key question.
You mentioned something earlier that always fascinates me, this idea that all the different cells in a plant, from a root hair to a huge trunk, they all come from one single cell, the zygote. How do they end up so incredibly different, both structurally and functionally? What drives that diversity?
That's the million-dollar question. And it brings us right to the heart of it: development. You see, development isn't just one thing. It's the sum total of two absolutely vital processes: growth and differentiation.
Growth and differentiation. Okay.
Right. And it's not just random changes happening. It's a very precise, highly ordered sequence of events unfolding throughout the plant's entire life. From that single zygote, the plant builds its complex body, systematically producing roots, leaves, branches, flowers, fruits, seeds, eventually leading towards senescence, which is biological aging, and finally, death.
Wow. So the diversity is all part of this ordered developmental plan.
Exactly. It's incredibly orchestrated. Now, the very first crucial step in this whole process is seed germination. A seed isn't just dormant, it's a tiny plant waiting. And it will only germinate when, and this is key, favorable conditions for growth exist in the environment.
Favorable conditions? Like what specifically?
We're talking about things like enough moisture water is crucial to hydrate the seed and get enzymes working. Then there's the right temperature range to, kickstart metabolism, not too cold, not too hot, and also adequate oxygen. Plants need oxygen for respiration, just like us, to get the energy for that initial growth spurt.
Okay, water, temperature, oxygen. Makes sense.
But what if those conditions aren't met? What if it's too dry or too cold?
Then it just waits.
Exactly. It enters a period of suspended growth or rest. We call this dormancy. Think of it like the plant hitting a pause button. It's a survival strategy, a waiting game.
Smart.
Very smart. And as soon as those favorable conditions return, maybe the rains come or spring arrives, the seeds resume their metabolic activities, break dormancy, and then growth happens. And it's crucial to remember, these developmental processes, they're governed by both intrinsic factors.
Internal factors, like genetics.
Yes, exactly. Internal factors like the plant's genetic makeup and internal chemical signals. And also by extrinsic factors.
External ones, like the environment.
Precisely. External factors like light, temperature, water, oxygen. We'll explore both types in more detail later, but the key point now is that both internal programming and external cues work together, tightly coordinated.
Okay, let's zoom in on growth itself. It's probably the most fundamental, most obvious characteristic of any living thing, right?
Yeah, things grow. But what is growth, biologically speaking? For your exams, NEET especially, you need this precise definition. Growth is an irreversible, permanent increase in size of an organ or its parts, or even of an individual cell.
Irreversible and permanent. Got it.
Yeah.
Keywords.
Absolutely crucial keywords. Once a leaf is fully grown, you can't shrink it back to a bud. It's a one-way street. And here's another critical point. This biological growth is always accompanied by metabolic processes.
Metabolic processes, the chemical reactions inside.
Exactly. Both anabolic, building things up, like proteins or cellulose, and catabolic, breaking things down to release energy, like respiration. And all these metabolic activities occur at the expense of energy. Growth isn't free. It costs the plant energy.
Okay.
So, for instance, when a tiny leaf bud expands into a big, mature leaf, that's growth. It requires energy to build new cells and expand existing ones.
Right, that makes sense. Energy intensive. So thinking back to what you said, if I soak a piece of dry wood in water, it swells up, it gets bigger. Is that growth?
Excellent question. Perfect for understanding the definition. No, that swelling is not biological growth.
Why not? It got bigger.
Because it's reversible, first of all. Dry the wood, and it shrinks back. It's just physical imbibition of water. And more importantly, there are no metabolic processes involved. No energy spent by living cells to build new protoplasm. It's just physics, not biology.
Okay, I see the distinction. Biological growth needs metabolism and is permanent.
Precisely. Now, what makes plant growth so special compared to, say, animal growth? Does a plant ever really stop growing?
Some trees seem to grow forever.
Exactly. That leads us to this unique feature. Plant growth generally is indeterminate. Plants have this amazing capacity for unlimited growth throughout their life. Think of those giant, ancient trees. Humans, we stop growing taller after adolescence.
Right. So what gives plants this ability?
It's all down to special regions called meristems. These are like permanent cell factories located at specific points in the plant body. Meristematic cells have this unique ability. They can divide and self-perpetuate. They keep dividing, making more of themselves and providing new cells for growth indefinitely.
So they don't run out.
Essentially, no. But here's the clever part. The cells they produce, the daughter cells, they soon lose the ability to divide. These cells then go on to mature, differentiate, and form the actual plant body, the permanent tissues.
Okay, so the meristem keeps dividing, but its products stop dividing and specialize.
You've got it. And this type of growth, where new cells are constantly being added by the meristem, is called the open form of growth.
Open form. Makes sense.
Now, you've likely learned about the root apical meristem and the shoot apical meristem.
Yes, at the tips of roots and shoots.
Correct. These are responsible for what we call primary growth. This is mainly about the elongation of the plants along their axis, making the stem taller, the root longer. But there's another type. In many dicots and all gymnosperms, you find lateral meristems.
Lateral, sideways.
Exactly. They appear later in life. The main ones are the vascular cambium and the cork cambium. Unlike the apical meristems, these lateral ones cause an increase in the girth or thickness of the plant parts where they're active. This is secondary growth.
So primary is length, secondary is width, like tree trunks getting thicker.
Perfect analogy. Primary growth builds width, secondary growth builds width and strength. If you picture those diagrams, maybe like figure 13.2 in your NCERT textbook, you can see where these different meristems are located.
Okay, that distinction between primary and secondary growth is clear. But how do we actually measure this growth? Is it just using a ruler? Or are there different ways, especially considering cell division versus cell enlargement? And are there any really impressive examples?
Good question, and very relevant for exams. Yes, growth is measurable, but it's not always simple. Fundamentally, at the cellular level, growth is about an increase in the amount of protoplasm, the living stuff inside the cell.
But that sounds hard to measure directly.
Exactly. Very difficult and impractical on a large scale. So instead, we measure some quantity which is more or less proportional to it, a proxy measurement. This gives us several parameters we can use. Increase in fresh weight, dry weight, length, area, volume, and even just counting the increase in cell number.
Different ways depending on what you're looking at.
Precisely. Let me give you some mind-blowing examples from your textbook. Consider a single maize root apical meristem, just the tip. It can produce more than 17,500 new cells per hour.
Wow, per hour.
Per hour. It's like a cellular assembly line going full speed. Here, growth is expressed as increase in cell number. Now, contrast that with a watermelon. The cells in a watermelon fruit can increase in size by up to 350,000 times.
350,000 times bigger. That's incredible.
Isn't it? That's not adding cells. It's the sheer increase in the size of the cell.
Yeah.
Pure enlargement. Or think about a pollen tube growing down towards the ovule. Its growth is mainly measured by its length.
Okay.
And for a typical flat leaf, like a mango leaf, growth is best captured by the increase in its surface area.
Right, how much bigger the flat surface gets.
Exactly. So you see, the parameter depends on the structure and the type of growth. Understanding which parameter fits which scenario is key for MCQs. Okay, moving on. Let's talk about the phases of growth. A plant cell or organ doesn't just grow uniformly. It goes through distinct stages. We generally divide this into 3 phases: meristematic, elongation, and maturation.
Meristematic, elongation, maturation. Okay.
The easiest way to visualize this is using a root tip, like in figure 13.3 of your text. Imagine tracking a cell's journey from the very tip upwards. First, right at the apex, the growth point, is the meristematic phase. This zone is all about cell division. It's the engine room. The cells here are specialized for division, rich in protoplasm, dense living material, large conspicuous nuclei, the control centers, and their cell walls are primary, thin, cellulosic, flexible.
Ready to divide and expand.
Exactly. And they have lots of plasmadonatal connections, little channels linking them for communication. Now, just proximal to this zone, meaning just behind it, away from the tip, is the elongation phase. Cells here aren't primarily dividing anymore. They're rapidly expanding. Key features: increased vacuolation. They develop large central vacuoles that fill with water.
Pushing the cell bigger from the inside.
Precisely. That water pressure drives cell enlargement, and as they expand, they deposit new cell wall material to strengthen the growing wall. This is where most of the length increase happens.
Got it. Elongation getting bigger.
Right. Then, further away from the apex, even more proximal, we find the maturation phase. Here, the cells have finished enlarging. Now they attain their maximal size and undergo wall thickening and protoplasmic modifications. They're differentiating.
Specializing for their final job.
Exactly. They're settling into their careers. Most of the tissues you studied earlier, xylem, phloem, parenchyma, they represent cells in this maturation phase, perfectly structured for their function.
Okay, that sequence from division to elongation to maturation makes sense. Now, what about growth rates? How fast does this happen? Are there different patterns we need to know for exams? And how do we express these mathematically?
Absolutely crucial area, this one. Growth rates and patterns are frequently tested, often with graphs or simple calculations. So, growth rate is simply defined as the increased growth per unit time. How much bigger does it get in, say, an hour or a day? Now, organisms can produce new cells in different ways, leading to different growth patterns over time. Let's look at the 2 main types. First, arithmetic growth. In this pattern, imagine a cell divides. Only one of the 2 daughter cells keeps dividing. The other one differentiates, matures, and stops dividing.
So only one branch of division continues.
Exactly. Like one worker trains a new worker, but the first one then goes off to do the actual job. The classic example is a root elongating at a constant rate.
Okay.
If you plot lengths against time for arithmetic growth, you get a straight line, a linear curve, just like in figure 13.5.
Linear increase, constant rate.
Precisely. And the formula is simple. LT is the initial length at time 0. Let's break that down. L is the length at time t. LUR is the initial length at time 0. And R is the growth rate or elongation per unit time.
Like adding the same amount each day. Simple interest.
Perfect analogy. Or adding a fixed number of bricks to a wall each day. Steady linear increase. Now, contrast that with geometric growth. This is much more common in biological systems, especially initially. In geometric growth, after a cell divides, both daughter cells retain the ability to divide, and they continue to do so.
Ah, so it compounds. Both keep dividing.
Exactly. It leads to exponential increase. But it typically follows phases. Initially, growth is slow, the lag phase, because the number of dividing cells is small. Then it accelerates rapidly, the log phase, or exponential phase. This is where growth is fastest, as the number of dividing cells explodes.
Makes sense. More cells dividing means faster overall growth.
Right. But in nature, resources aren't infinite. So eventually, maybe due to limited nutrients or waste buildup, the growth slows down, leading to a stationary phase. If you plot a growth parameter against time for geometric growth, you get that classic sigmoid or S-curve. Look at figure 13.6.
The S-shaped curve, I've seen that.
It's incredibly important. This S-curve is characteristic of living organisms growing in a natural environment. It's typical for pretty much all cells, tissues, and organs of a plant under normal conditions.
Wow. Universal for natural growth.
Largely, yes. Now, the exponential part of this curve can be expressed mathematically. W1 is the final size, weight, height, number, whatever. WE is the initial size. T is time. T is the base of natural logarithms, about 2.718. And here is the relative growth rate. It's also a measure of the plant's efficiency in producing new material, sometimes called the efficiency index.
Relative growth rate, efficiency index, okay.
So the final size W1 depends heavily on the initial size W0, and this compounding rate R over time T.
Like compound interest, the growth itself starts generating more growth.
Exactly. That's the perfect analogy for geometric growth. Exponential increase, then leveling off.
Okay, arithmetic is linear, geometric is exponential, leading to an S-curve. Got it. But you use the term relative growth rate for R in geometric growth. How exactly do absolute and relative growth rates differ? Why is this distinction important? Sounds like something that could trip me up on an exam.
You're right to focus on this. It's a common point of confusion, but really important conceptually. When we compare growth quantitatively, we use these two measures. Absolute growth rate is straightforward. It's the total growth per unit time. Simple measurement. If a leaf grows by 5 centimeters in a week, the absolute growth rate is 5 centimeters per week. Raw increase.
Just the amount it grew.
Okay.
Yeah.
Relative growth rate is the clever one. It's the growth per unit time expressed on a common basis, e.g. per unit initial parameter. Basically, it's the growth relative to the starting size. It tells you about the efficiency or proportional increase.
Efficiency, proportional. Okay. How does that work?
Let's use that classic example, often shown like figure 13.7. Imagine two leaves, A and B. Leaf A starts small, say 5 centimeters away. Leaf B starts much larger, say 50 centimeters away. Over 1 week, both leaves increase their area by the exact same absolute amount, 5 centimeters around.
Okay, both grew by 5 centimeters.
Right. So leaf A goes from 5 to 10 centimeters of A1. Leaf B goes from 50 to 55 centimeters of A1. Now the crucial question, which one had a higher relative growth rate and why?
Leaf A doubled its size. Leaf B only increased by 10%, so leaf A.
Exactly. Leaf A's relative growth rate is 5 centimeters or gain, 5 centimeters or initial equally 1 or 100%. Leaf B's relative growth rate is 5 centimeters gain, 50 centimeters or initial equals 0.1 or only 10%. So even though the absolute growth was the same, Leaf A's relative growth rate was much higher because the increase was a huge proportion of its starting size.
Ah, I see. Relative rate shows how fast it's growing compared to how big it was. That's a key difference.
Precisely. It measures the efficiency of growth. Very important concept for comparing different plants or organs.
Makes perfect sense now. Okay, so finally, wrapping up our discussion specifically on growth, what are the absolute essential conditions a plant needs? What environmental and internal factors are critical?
Excellent summary question. These are the non-negotiables for plant growth. We need water, oxygen, nutrients, the right temperature, and certain environmental signals. First, water. Absolutely fundamental. Cell enlargement, which drives most growth, depends on turgidity, that internal water pressure.
Turgor pressure pushes the cell wall out.
Exactly. It drives extension growth. No water, no turgor, no growth. Plus, water is the medium for all enzymatic activities needed for metabolism.
Can't live without water. Got it.
Second, oxygen. Plants make oxygen in photosynthesis, but they need it for respiration to release metabolic energy. That energy, ATP, fuels everything: cell division, nutrient transport, synthesis. No oxygen, no energy, no growth. Think waterlogged roots, they suffocate.
Right. Respiration needs oxygen.
Third, nutrients. Macro and micro elements from the soil. These are the building blocks for protoplasm and sometimes sources of energy. Nitrogen, phosphorus, potassium, macro, iron, zinc, micro, all essential. A deficiency in just one can halt growth.
Building materials, okay.
Fourth, temperature. Every plant has an optimum temperature range for growth. Enzymes work best within this range. Deviate too much, too cold, or too hot, and metabolic processes slow down or enzymes get damaged, growth stops, survival is threatened.
Goldilocks zone for temperature.
Pretty much. And lastly, environmental signals like light and gravity. Light isn't just for photosynthesis, it directs growth, phototropism, and flowering, photoperiodism. Gravity ensures roots go down, shoots go up, gravitropism.
So the environment guides growth, too.
Absolutely. All these factors, water, oxygen, nutrients, temperature, signals, are indispensable and interact complexly.
Okay, so we've thoroughly covered growth. Now let's shift gears slightly to these fascinating cellular journeys. Differentiation, dedifferentiation, and redifferentiation.
Right, how cells change and specialize. Exactly. Let's start with differentiation. Cells, or the meristems, are initially quite generic, but then they differentiate and mature to perform specific functions. This process of becoming specialized is differentiation. It involves structural changes in both cell walls and protoplasm, tailored to the final job.
Structure matching function again.
Always. Take a tree element. a cell forming xylem for water transport.
Yeah.
During differentiation, it undergoes drastic changes. It actually loses its protoplasm, becoming a hollow tube.
It dies.
Functionally, yes. The cell itself dies to become a deficient pipe. And it develops strong, elastic, lignocellulosic secondary cell walls. Why? To handle the tension of pulling water up long distances without collapsing. Amazing engineering.
Wow, that's extreme specialization. So is differentiation always final? Once a cell becomes a tertiary element, is that it? Or can plant cells change their path?
That's a brilliant question. And the answer is one of the amazing things about plants. No, it's not always a one-way street. Plants show dedifferentiation. This is where living differentiated cells that have stopped dividing, can regain the capacity of division under certain conditions.
They become meristematic again.
Exactly. They despecialize. A prime example is the formation of interfascicular cambium and court cambium from fully differentiated parenchyma cells. Mature parenchyma can dedifferentiate to form these crucial secondary meristems.
Incredible flexibility. So what happens after they dedifferentiate?
Good follow-up. Once these new meristems, like the cambium, are formed and dividing, the new cells they produce then once again lose the capacity to divide but mature to perform specific functions. This process of respecializing after dedifferentiation is called redifferentiation.
Dedifferentiate, divide, then redifferentiate. A cycle.
In a way, yes. Think about woody dicots. The secondary xylem and phloem from the vascular cambium, or the cork from the cork cambium, these are products of redifferentiation. Now, let's connect this back to open growth. We can say that even differentiation in plants is open.
What does open differentiation mean?
It means that cells arising out of the same meristem can end up having different structures at maturity. Their fate isn't predetermined solely by origin, and crucially, the final structure is also determined by the location of the cell within the plant body. Position matters immensely.
Location determines fate. Give me an example.
Okay, think of the root tip again. Cells positioned away from the very tip, but near the meristem, differentiate into protective root cap cells. But cells pushed towards the periphery, the outside, mature as epidermis, the outer skin. Same origin, different location, different fate.
Wow. So context is everything.
Precisely. Think about vascular bundles, too. Central cells become xylem, peripheral ones become phloem. Location, location, location.
That clarifies it beautifully. Now, you mentioned parenchyma cells dedifferentiating. I remember reading about tumors in plant tissue culture. How does that relate to this? Is a callus a tumor?
Excellent connection, yes. Relates directly. A tumor in plants, like in animals, is basically abnormal, uncontrolled cell proliferation. In plant tissue culture, we actually exploit dedifferentiation. We take differentiated cells like parenchyma, put them in a special medium, and under controlled lab conditions, we make them divide again. That's dedifferentiation in action.
Right, forcing them back into division.
Yes. And this massive dividing, undifferentiated cells they form is called a callus. In a way, it is like a controlled tumor in vitro uncontrolled proliferation. But the key is we can then control it. By changing the hormones in the medium, we can make the callus cells redifferentiate into roots, shoots, eventually a whole new plant.
So tissue culture relies on the cycle of dedifferentiation and redifferentiation. Amazing.
It really is. It showcases the incredible developmental flexibility of plants. Okay, let's zoom out again now to the big picture, development. This term covers everything. Development includes all changes an organism goes through during its life cycle, from germination of the seed to senescence and death. The whole journey.
The entire life story.
Exactly. We can visualize the sequence for a single plant cell, like in figure 13.8. It starts with a meristematic cell, then cell division, then plasmatic growth, making more protoplasm, then expansion elongation, getting bigger, followed by maturation, then differentiation, specializing, leading finally to senescence, aging, and death.
That sequence applies broadly to tissues and organs, too.
Yes. This sequence is a general pathway applicable to tissues, organs, and the whole plant.
It sounds very ordered, but I've heard plants can be plastic, like having different leaf shapes on the same plant. How does plasticity fit in? What's better for...
Great point. Plasticity shows that development isn't completely rigid. Plants are adaptable. Plasticity is the ability of plants to follow different pathways in response to environment or phases of life to form different kinds of structures. Flexibility in form.
Adapting their structure. Okay.
And heterophily is a perfect example. Hetero means different. Phily means leaves. So different leaves on the same plant.
How does that happen?
Two main ways. In plants like cotton, coriander, and larkspur, the leaves of the juvenile plant are different in shape from those in mature plants. Young cotton leaves might be lobed, mature ones less so. That's developmental plasticity.
Shape changes with age.
Right. But in buttercup, an aquatic plant, you see environmental plasticity. Leaves produced underwater are finely dissected, ribbon-like.
Adapting to water flow.
Exactly. The leaves produced above water are broad and lobed for air and sunlight. Same plant, different environment, different leaf shape. Check figure 13.9, it shows this clearly.
Wow. So heterophilia is a clear example of plasticity.
Definitely. It shows development isn't strictly predetermined, but responsive. So to wrap up this part, growth, differentiation, and development are very closely related events. Development is broadly the sum of growth and differentiation. And remember, all of this is controlled by both intrinsic factors, genetic and chemical signals like hormones, and extrinsic factors, light, temperature, water, etc.
Intrinsic and extrinsic controls working together.
Always. It's a complex interplay. Right, now let's get into what is arguably one of the most exciting and certainly very high-yield sections for your exams, plant growth regulators, or PGRs.
Ah, the plant hormones. Yes, this sounds crucial.
Absolutely. Think of PGRs as the chemical messengers, the orchestrators directing almost everything in a plant's life. They're generally small, simple molecules of diverse chemical composition, small size, huge impact. You'll also see them called plant growth substances, plant hormones, or phytohormones.
Different name, same thing, basically.
Essentially, yes. And chemically, they're quite varied. We have indole compounds like IAA, indole 3-acetic acid, adenine derivatives like kinetin, then derivatives of carotenoids like ABA, pittusic acid, terpenes like GA3, gibberellic acid, and even a gas, ethylene, C2H4.
Such different chemicals doing similar jobs.
Similar types of jobs regulating growth, but with very specific effects due to their different structures. We can group them broadly based on function. First, plant growth promoters. These are involved in promoting activities like cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, seed formation. The main examples: auxins, gibberellins, and cytokinins.
The go signal.
Exactly. Then we have plant growth inhibitors. These are crucial for responses to wounds and stresses, biotic and abiotic, and for growth inhibiting activities like dormancy and abscission, shedding leaves, fruits. The main one here is abscisic acid, ABA. Interestingly, ethylene, the gas, can kind of fit in both groups, but it's largely an inhibitor, especially regarding aging and shedding.
Okay, promoters, auxins, gibberellins, cytokinins, and inhibitors, ABA, largely ethylene. That's a helpful grouping. So how are these actually discovered? You said small, simple molecules. Did scientists set out looking for them, or was it more accidental? The discovery stories are always fascinating.
They really are. And you hit the nail on the head. The discovery, each of the 5 major groups, was remarkably largely accidental, often stumbled upon while investigating something else entirely. These stories are classic exam material, too.
Accidental discoveries. Cool. Let's hear them.
Okay, starting with auxins. It all began with Charles Darwin and his son Francis. They were studying phototropism, why plants bend towards light. They used coleoptiles of canary grass, that little sheath covering the young shoot. They noticed the bending happened towards light.
Right, the classic experiment.
Through clever experiments covering different parts, they concluded the tip of the coleoptile was the site of transmittable influence that caused the bending, even though the bending itself was lower down. A signal was being sent.
The tip senses the light and tells the lower part to bend.
Precisely. Years later, FWN actually isolated this chemical influence from oat coleoptile tips and named it auxin. You often see diagrams like figure 13.10 illustrating this.
Okay, auxins from phototropism studies. What about gibberellins?
Gibberellins have a strange origin story, the Botany, or foolish seedling disease in rice in Japan. Infected seedlings grew ridiculously tall and spindly, then fell over. Foolish. It was caused by a fungus, Gibberella fujikuroi.
A fungus causing tall growth. Weird.
Very. In 1926, E. Kurosawa found that treating healthy seedlings with sterile filtrates of the fungus is just the liquid the fungus grew in. No fungus cells cause the same symptom.
Ah, so the fungus released a chemical.
Exactly, a growth-promoting substance. Later, these active substances were identified as gibberellic acid.
Okay, gibberellins from a rice disease fungus. Next, cytokinins.
Cytokinins came from plant tissue culture work. F. Skoog and his team were growing tobacco stem segments. They found that the callus, the cell mass, only proliferated vigorously if the auxin medium was supplemented with things like extracts of vascular tissues, yeast extract, coconut milk, or DNA.
Strange additives? What was in them?
Something promoting cell division, or cytokinesis. Miller and colleagues eventually isolated and crystallized the active substance from degraded DNA in 1955. They called it kinetin.
Kinetin from DNA. Interesting. What about ABA, abscisic acid?
ABA's discovery was a convergence. In the mid-1960s, 3 independent research groups reported purifying 3 different inhibitors: inhibitor B, abscission 2, and dormin.
3 different inhibitors found at the same time.
Yes, investigating different things like dormancy and shedding, but later it turned out all 3 were chemically identical.
Wow. They all found the same molecule.
Exactly, so it was unified under the name abscisic acid, ABA, science converging.
Cool. And finally, ethylene, the gas.
Ethylene's discovery goes back to a practical observation in 1910 by H. Cousins. He confirmed that ripened oranges released a volatile substance, a gas that hastened the ripening of stored unripened bananas nearby.
Ah, the one bad apple spoils the bunch idea, but with oranges and bananas.
Pretty much. That volatile substance was later identified as ethylene, a simple gas acting as a hormone.
Amazing how many came from observations or accidents. Okay, now that we know their history, let's really dive deep, the physiological effects and practical uses of each one. This feels like the absolute core for exams.
You are absolutely right. This section is critical. Let's break down each of the 5 major PGRs and detail their functions, their applications in agriculture and horticulture. Pay close attention here. First, auxins. Remember, auxin means to grow. First isolated from human urine, interestingly, the main natural one is IAA, indole 3-acetic acid. Another natural one is IBA, indole butyric acid. But we also have important synthetic ones like enedol A, naphthalene acetic acid, and 2,4-D, 2,4-dichlorophenoliacetic acid.
Natural and synthetic types, where are they made in the plant?
Primarily in the growing apices of stems and roots, the very tips. From there, they're transported to exert their effects. Both types are widely used practically. Okay, functions and applications, huge one. They initiate rooting in stem cuttings. Essential for plant propagation. Treat a cut stem with auxin, you get roots.
Making new plants from cuttings, very useful.
Very. They also promote flowering, for example, in pineapples, helping synchronize the harvest. They have a dual role in shedding. They help prevent fruit and leaf drop at early stages, keeping young ones attached, but they promote the abscission of older mature leaves and fruits. Cleaning house, essentially.
Prevents young fruit drop, promotes old fruit drop.
Okay. Now the super important concept. Apical dominance. The growing apical bud inhibits the growth of the lateral axillary buds below it.
So the main stem grows tall, side branches don't.
Exactly. If you remove the shoot tip, decapitation, the lateral buds start growing vigorously, like in figure 13.11.
Yes.
This is widely applied in tea plantations. Constant pinching promotes bushiness for more leaves and in hedge making. Auxins also induce parthenocarpy fruit development without fertilization. Think seedless tomatoes.
Seedless fruit. Okay.
And they're used as herbicides. Specifically, 2,4-D is widely used to kill dicotyledonous weeds, broadleaf weeds, without harming mature monocotyledonous plants, like grasses. Perfect for weed-free lawns.
Selective weed killer. Very handy.
Indeed. They also control xylem differentiation and help in cell division. Auxins are real master regulators, especially for primary growth. Next up, gibberellins, GAs. Another group of promoters. Over 100 known types, GA1, GA2, GA3, all are acidic. GA3 is the most studied.
More than 100. Wow. What do they mainly do?
Their standout effect is causing an increase in length of axis, making things longer or taller, applied to increase the length of grape stalks, making bigger bunches. Also used to make apples elongate and improve shape.
Longer grapes, shapelier apples.
They also delay senescence, slowing down aging. Practical use. Fruits can be left on the tree longer, extending the market period.
Delaying harvest, useful for farmers.
Very. GA3 is also used to speed up the malting process in brewing. Faster beer production. And here's a big one for agriculture. Sugarcane. Spraying sugarcane with GAs increases the length of the stem, which is where the sugar is stored. This can increase yield by as much as 20 tons per acre.
20 tons per acre increase. That's huge. Definitely remember that number.
Absolutely. For forestry, spraying juvenile conifers with GAs hastens maturity, leading to early seed production. Good for breeding. And they promote bolting, that rapid stem elongation, before flowering in rosette plants like beets and cabbages.
Making the stem shoot up.
Exactly. So GAs are primarily the elongation or tall growth hormones. Third, cytokinins, named for their effect on cytokinesis cell division. Remember, quinoa was found in herring sperm DNA, not natural in plants. But natural ones like zeatin were found later in corn kernels and coconut milk.
Where are natural cytokinins made?
In regions where rapid cell division occurs. Root serieses, developing shoot buds, young fruits, active growth zones, their functions. They help produce new leaves, promote chloroplasts in leaves, making them greener, more efficient, stimulate lateral shoot growth, and adventitious shoot formation.
Promoting leaves, chloroplasts, side shoots, making the plant bushier.
Yes, and crucially, they help overcome apical dominance. They act antagonistically to auxins here. Auxins inhibit side buds. Cytokinins promote them.
Ah, the balance between auxins and cytokinins controls branching.
Precisely. They also promote nutrient mobilization, directing nutrients to growing areas, which helps delay leaf senescence, keeps leaves working longer. So cytokinins are the cell division and branching hormones. Fourth, ethylene, the unique simple gaseous PGR, synthesized mainly by tissues undergoing senescence and ripening fruits.
The ripening gas, what else does it do?
Lots. It causes horizontal growth of seedlings, swelling of the axis, and apical hook formation in dicot seedlings emerging from soil. Protects the tip. It strongly promotes senescence and abscission of leaves and flowers. Signals aging and shedding. And yes, it's highly effective in fruit ripening. It triggers that respiratory climactic, the burst of respiration during ripening. Remember that term?
Respiratory climactic, got it. Increased respiration during ripening.
It also breaks seed and bud dormancy, initiates germination in peanut seeds, promotes sprouting of potato tubers, waking things up.
Breaking dormancy too, interesting.
And in plants like deep water rice, it promotes rapid internode to petiole elongation to keep leaves above water, a survival mechanism. It also promotes root growth and root hair formation, increasing absorption surface.
So quite versatile. Ripening, aging, breaking dormancy, elongation, root growth.
Very versatile. Used commercially to initiate flowering and synchronize fruit set in pineapples and induce flowering in mango. It's one of the most widely used PGRs in agriculture, often applied using aethifon.
Aethifon, what's that?
Aethifon is a liquid that's absorbed by the plant and releases ethylene slowly inside. Very convenient. Uses of aethifon: hastens fruit ripening in tomatoes and apples, accelerates abscission for thinning cotton, cherry, walnut, and it promotes female flowers in cucumbers, increasing yield.
Wow. Ethylene is complex aging and ripening, but also growth and stress responses.
It is. Think of it as the ripening and aging hormone, but with important roles in stress adaptation too. Finally, abscisic acid, ABA. Remember, discovered for its role in regulating abscission and dormancy. It generally acts as a plant growth inhibitor and an inhibitor of plant metabolism. Slows things down.
The main stop signal.
Largely, yes. It inhibits seed germination. It stimulates the closure of stomata, helping conserve water under stress. It increases tolerance to various stresses: drought, salt, cold. That's why it's called the stress hormone. Very important name to remember.
ABA equals stress hormone. Stimulates stomatal closure. Increases stress tolerance.
Exactly. It's crucial for seed development, maturation, and dormancy. Inducing dormancy helps seeds withstand desiccation and unfavorable conditions.
Puts seeds to sleep safely.
Good way to think of it.
Yeah.
And remember, in most situations, ABA acts as an antagonist to GAs, especially regarding dormancy. GAs break dormancy. ABA induces it.
The push-pull between ABA and gibberella.
Yes. So ABA is the plant's stress manager, the dormancy switch. Okay, so wrapping up PGRs. It's crucial to see they rarely act alone. The roles can be complementary, working together, or antagonistic, opposing each other. They can be individualistic, one dominates, or synergistic. Combined effect is greater than the thumb.
Complex interaction.
Very. Many events like dormancy, abscission, senescence, apical dominance involve the interplay of more than one PGR. It's about the balance. And remember, PGRs are just one type of intrinsic control. They work alongside genomic control, genes, and extrinsic factors, environment. In fact, many environmental cues like temperature and light control plant growth via PGRs. Think vernalization, flowering timing, seed germination environment signals, hormones execute.
So the environment talks to the hormones to control the plant.
Beautifully put. It's a fully integrated system. So to quickly recap our entire deep dive today on plant growth and development, we started with growth. Irreversible, permanent, metabolic, often indeterminate in plants due to meristems causing that open form of growth. So that's measurable in many ways: cell number, maize root, cell size, watermelon, length, area.
Right, those amazing examples.
Then the phases: meristematic, elongation, maturation, the growth rates, arithmetic, linear versus geometric, S-curve, and that crucial difference between absolute and relative growth rates.
Relative growth rate showing efficiency, got it.
And the essential conditions: water, oxygen, nutrients, temperature, light, gravity. Then we explored differentiation, specializing, like tracery elements, dedifferentiation, regaining revisionability, and redifferentiation, respecializing. We saw how location matters, open differentiation, and how this relates to tissue culture, callus.
And development as the whole life cycle, including plasticity and heterophyly.
Exactly. Development as the sum of growth and differentiation, showing adaptability. And finally, the big one, plant growth regulators. Auxins, rooting, apical dominance herbicides, gibberellins, elongation, bolting, sugarcane yield, cytokinins, cell division, branching, delaying senescence, ethylene, ripening, abscission, stress responses, the gaseous one, and abscisic acid, dormancy, stress hormone, stomatal closure. We covered their discoveries, effects, applications, and complex interactions.
That was incredibly comprehensive. So much detail.
It is a dense chapter, packed with concepts vital for your boards and NEET. My strong advice, revise this again thoroughly. Pay very close attention to the specific examples and applications of each PGR. Those are prime exam questions. Definitely practice past year questions on this topic. It really helps solidify understanding. And remember, if any doubts remain after you revise, please do reach out for clarification. Keep learning, keep growing.